Hi, I am Jackie Goodrich. Today I'm going to tell you about my research at the University of Michigan School of Public Health. Humans around the globe are exposed to chemicals and other stressors on a daily basis. We come into contact with these chemicals through consumer products, air, water, and even food. In the United States alone, tens of thousands of chemicals are produced and sold. Whether all of these chemicals are safe for chronic exposure, especially among vulnerable populations, is not well known. The central goal of my research is to identify chemicals that have a long-term impact on health. In particular, I study the effects of exposures in epidemiological cohorts of mothers and children or of workers. Why children and adult workers? While these two groups are very different, it is imperative for risk assessment and policy making that we understand the effects of chemical exposures on both. They are each vulnerable to toxicity. Children are rapidly developing and have unique exposure sources. Chemicals can disrupt these sensitive periods of development, leading to lifelong changes in health. Certain worker groups are chronically and highly exposed to certain chemicals and other hazards. We need to know if these higher doses of exposure are safe in the long run. How can we know whether chemical exposures lead to long-term health impacts? Their effects are often subtle and remain undetected for years before manifesting as clinical disease such as cancer or cardiovascular disease. Through my research, I investigate whether epigenetic changes from chemical exposures lead to subtle alterations in normal biology that lead to disease. Epigenetics encompasses modifications to DNA and chromatin that control the expression of genes but do not change the genetic sequence. The entire genetic sequence or genome can be likened to the keys of a piano. The keys of all pianos encode the same musical notes. Likewise, all the cells that make up an individual person contain a copy of essentially the same genetic code. When the piano is played, there are nearly infinite possibilities for musical expression. Similarly, the epigenome controls the expression of genes and is exquisitely tuned in every cell type and in every stage of development and aging to express certain genes in certain tissues. Unfortunately, altered epigenetics can disrupt development in typical biological processes, leading to disease. Environmental exposures, including chemicals, diet, and even stress, have been shown to alter the epigenome. Epigenetic modifications are heritable across cell divisions, meaning the impact of exposure can be propagated over time, even if exposure stops. This is one reason why children are so vulnerable to impacts from the environment. Many of my research studies involve characterizing common exposures, such as to lead, phthalates, and phenols, in pregnant women and following up their children to assess whether these exposures are associated with birth outcomes, altered growth trajectories, childhood obesity, and more. In Mexico City, our team has followed the same children from gestation until early adulthood. However, such longevity is not feasible for most human studies. In other studies, I profile epigenetic modifications in samples from the kids collected at birth or in early childhood to infer whether exposures cause subtle biological changes that have implications for lifelong health. I also participate in studies of adult workers to infer whether occupation-specific exposures have effects on the epigenome and whether these subtle changes increase their risk for disease. Overall, my research addresses a prominent public health issue. I investigate whether chemical exposures increase risk for disease, in part through epigenetic changes. This knowledge can be used to inform risk assessment for chemicals on the market. Furthermore, epigenetic data can inform prevention and intervention strategies. Epigenetic changes linked to disease can be developed into biomarkers that predict future disease, or they can inform therapeutic strategies to disrupt toxicity and disease. Thanks for listening.